Hi, everyone. Thank you for joining. My name is Anna Maria Istrate, and I am a senior research scientist at, excuse me, Chan Zuckerberg Initiative. And I'm excited to share a little bit today about some of the work we're doing at CZI in using natural language processing to extract various type of information from biomedical research articles. And I realize not everyone might be familiar with the biomedical field, so feel free to interrupt me at any point if you need any clarifications or anything like that. I just want to start by giving a very short overview of what we do at CZI. So CZI is a tech-focused philanthropy, and in particular, I work on CZI Science. So CZI Science is a focus area of CZI, and our North Star is really supporting the science and technology that will make it possible to cure, prevent, or manage all diseases by the end of this century. And one of our core values is that of open science. So we believe in the universal and immediate open sharing of all scientific knowledge, processes, and outputs. And some of the projects I'll be talking about today, in fact, all of the projects I'll be sharing today are in support of our open science program at CZI. So I want to start very broadly kind of talk about, transformer models and how we landed on the models we're using currently. So maybe most of you here are already familiar with this. If so, I apologize. But transformer models have, as I'm sure most of us are aware of, became really popular, mainly used in natural language processing, also computer vision. At the very core, they really are these deep learning models that are based on the idea of self-attention. So the main idea here is that you want to understand the current word given its context in the input sequence. They have been introduced a while back. So first introduced in 2017 in this paper, Attention is All You Need. And there's this example that I really like. You have the sentence, the animal didn't cross the street because it was too wide. So when you want to compute a representation for the word it, in this case, basically you need to know what other words to attend. As humans, we're able to say that, well, it refers to the street because the street was too wide. So this is kind of this core of self-attention. Once we had this transformer model architecture, we've seen this boom in the number of large language models and that are based on transformers that have been developed. So things like BERT, GPT, Electra, Roberta. And there's a lot of enthusiasm right now with things like ChatGPT, but these models really have been around for a little bit of time, like a few years now. So they really have this high performance on a variety of tasks. So if you're looking at things like NER, question answering, summarizations, other types as well, because they are pre-trained on these huge amounts of text, huge corpora. So they learn to capture these complexities in natural language really, really well. And the way we're using them, and I think most people are using them, is, you take these pre-trained models and you fine-tune them on your particular downstream task. So you take a model like BERT that has been pre-trained on one of these huge corpora. So for BERT, it would be like books corpus, English Wikipedia, and then you're not every time you want to use this model, you're not pre-training it again and again. You're just fine-tuning it on your particular task. So we use these models a lot to fine-tune them for our own tasks as well. And one of the challenges that we run into is that of specialized domains. So we work a lot with biomedical research articles. And the biomedical domain has a different word distribution than general domains. So that means that these representations that you're learning by these models from more general natural language domains are not necessarily capturing the complexities in specialized text. So for biomedical field, we care a lot about things like genes, cell lines, diseases, whatever that might look like. And what that means is that we want to kind of have more specialized models that are tailored to the biomedical field. So pre-training on domain-specific text has actually been shown to improve accuracy on downstream tasks, at least in the biomedical field. And we are lucky in a sense that we do have a lot. The biomedical field has large amounts of data that are openly accessible that can be used for pre-training. So we have, excuse me, so we have something that's called PubMed, that is a repository of titles and abstracts of research articles. But we also have repositories that give you access to full text research articles that you can use and pre-train. So we have two in particular we're using. It's called PubMed Central and Semantic Scholar. And these repositories contains millions of open access scientific articles. So how the biomedical field has adapted and using these models is you take these model architectures and then just pre-train them on these specialized corpora. So you start with BERT and end up with things like models like BioBERT that's been pre-trained on PubMed abstracts and these full text articles. SciBERT, it's trained on Semantic Scholar full text papers. So these are more scientific research articles, also contain papers in computer science. So you have these model variations. You go from Electra to BioElectra, or Roberta to Biomed Roberta. So one of the things that we are interested in, that kind of like ended the NLP overview portion, moving on into why, what are the things we care about as funders. Again, we are a philanthropy. We want to be able to recognize impactful outputs. So we want to be able to recognize what are impactful data sets, what is impactful software, what are these outputs that make the biggest impact on science. And we also want to identify impactful authors. So whether these are software developers or data set creators. So we're able to keep investing in them. The authors themselves, and this might be someone who developed an open source software or created a data set, they want to know their own impact. So you want to understand how are you being used in science or across science, who's using you and why. And you also want to get access to funding opportunities and things like citations or usage in science downloads. These are all things that matter for getting these funding opportunities. Researchers themselves, they just want to discover what data set can I use for my particular problem? What software should I use for my particular problem? What software are other people using? So this brings us to kind of the second challenge we have, which is a challenge for us, but it's a good thing, is that there's a lot of biomedical research being done, which is great because people are tackling lots of really impactful problems. But it's estimated you have around 3,000 to 5,000 biomedical research articles published daily. So then when you want to kind of extract this information, what's the impactful software or data set, it's really, you can't do it manually. It's there's no way to do that manually. And to give you a sense of some of the corpora we work with, we have an in-house corpus at CZI that has around 11 million papers. And one of the open access corpora available has around 3.8 million papers. So then we need some types of automated solutions. And this is where natural language processing comes into play in order to identify these kind of research outputs. So now I want to talk and go more deeply into three of the projects we've developed at CZI in collaboration with partners. And the first one I want to talk about is building the open global data citation corpus. What does that mean? In this project, we are looking to surface all paper data set links from scientific articles, and we want to make this data openly available to researchers. This is a project in partnership with some of our collaborators at DataCite. So, I mentioned we're interested in these research outputs, and data is a critical research output, and the biomedical field is becoming increasingly data-driven. So people are, really interested in and kind of taking a look at what data is being used, how it is being used, what are the impactful data sets. So we want to be able to discover it and surface it. The challenge is that there is no main repository right now that gives you, these links between what paper and what data set are using. There's no papers with code equivalent for the biomedical field or something like that. So, and a lot of the times why that happens is because, there have been efforts, but data is just not properly cited. So in an ideal world, you would have a reference list. So when you write a paper, you'd go and put data in your reference list, but authors just don't do that in practice. Journals have also different norms for like publishing data. So it's a really challenging task. And then what do we mean? However, the majority of these data sets are mentioned in the full text of papers. So authors will mention the data they're using in the full text of a paper somehow, which means that we can go and extract it straight from the source. So what do we mean by data? Usually it looks like an identifier in a particular database. So we have these examples here, GSC 40279. This is actually a mention of a data set in the geo repository. So this is what we're trying to identify. And we have narrowed down to 46 different databases we're interested in. The thing is that these identifiers, they're not straightforward to extract. So there's a lack of common naming conventions. So you can have something like that starts with AB, and then it's something, some sequence of numbers that could belong to maybe 20 different repositories. So you can't really use things like regular expressions or some other more rule-based approaches. And here we have, again, some examples of how these mentions appear in papers themselves. So in the first one, we have an example of authors mentioning they downloaded the data from this database, and then they point to the identifiers of those data sets. And then we want to be able to extract the data sets themselves, so these GSC accession numbers, as well as knowing that they belong to the geo database. And we want to do that because without knowing the database, these are just strings, and we can't do much with them because we want to be able to link them to something. So, you want to be able to access these data. So a string doesn't necessarily give you that. In the second example, we have we can see how an author mentioned how the data associated from their study was deposited in this Mendeley data repository, and they provide the accession number. So in this case, we want again to be able to extract a link between this paper and this DOI identifier. So one of the very first things we do when we tackle a problem like this is aligning on entity's definition. And this is actually more complex than it might look at first. So, what do we mean by data set? What do we include when we are talking about data sets? Do you include things like figures or, presentations? If you have protein sequences, does that constitute data? So it's actually a pretty challenging thing to do. And we do have a curation team. That's a team of biomedical experts that we usually collaborate with on these entity definitions. And that's very important for when we're building the models themselves as well. So how we're tackling this in practice, we abstract this as a named entity recognition task. So we are using these BERT models. We're fine-tuning them on this NER task. So just very briefly, we have a sentence, and we have an example here. All the sequences in raw FASTQ format have been submitted to this repository and so on. We're passing this sentence through the BERT tokenizer. We're tokenizing, that gives us the tokens. We're passing that through the BERT kind of like module. And this is the fine-tuning part where we have this linear classification layer where we are classifying each of the tokens as being either a part of a data set, and this data set, we're predicting both the fact that this is a data set and the repository belongs to. And then we're mapping these tokens back together into the corresponding words. So as mentioned, we're interested in 46 different classes, and we have one corresponding class to each of the databases we're interested. So our tags are basically being able to say, okay, this is a data set in bioproject repository, or this is a data set in this Array Express repositories. So these are the repositories we're interested in. We are using Hugging Face for this, which makes it very easy to load all of these models and kind of compare them as well. So we've experimented with multiple models, some of those pre-trained on biomedical text I was talking about. Our best model actually achieved pretty impressive results. We have a precision of 94%, recall 98, and F1 score of 96%, which is really good. Our training data has been curated with the help of our curation team, and it comes from 23,000 papers spanning 123,000 sentences. And for some examples here, the first one is quite interesting because the model, you're seeing these GSM identifiers, and the model is learning, is able to predict that these are data sets coming from the geo database. And you can see that there's nowhere in the sentence that geo database is actually mentioned, which is quite interesting. Second one is an example of identifying a protein sequence from a repository called GenBank. And the third one, we can see that we have these two separate identifiers, one from BioProject and one from BioSample. These are just different repositories, and the model is able to distinguish them appropriately. The first one being from BioProject and the second one BioSample. So our full pipeline, we have this model, and once we have the trained model, we run it on our corpora. So as I mentioned before, we have a full text corpus in-house at CZI, and then we are also running the models on open access corpora. There's a repository called EuroPMC we're using for that. Once we get these data set mentions as well as the database they belong to, we are able to create these links. So then we're making these links between papers and data sets available to the community. So now I want to move to the second project we've been working on at CZI, and this is building the Global Biodata Resource Inventory project. It's in collaboration with our partners at what it's called the Global Biodata Coalition. And really what they're interested in is in developing a sustainable financial support for the global biodata infrastructure. And in order to do that, we want to build a comprehensive inventory of life science data resources. So naturally, you might be asking, what's a biodata resource? What are we looking for? And similar to before, entity definitions for us is very important. I think for simplicity, you can think of this as a repository where people go and deposit or download their data. So it's an online source that hosts all types of data related to life sciences. So what we're interested in doing here is we want to extract all of these databases really from papers, because we want to build this comprehensive inventory. So we want to know everything that's out there. There's a few that we know exist. So there's a few popular databases, but we don't know all of them. So the goal here is really to be able to create this, network between these databases. So here. And these databases usually have a full name. So in this case, we have an example. It's called Functional Analysis Tools for Post-Translational Modification, and also an acronym. And in our case, and in this case, it's FAT-PTM. And we want to be able to extract both. We are also abstracting this as an NER task. And our workflow here, we actually have two ML models. So we start with the title, with the paper. We only have access in this case to the title and abstract of a paper. And then the first model is a paper classification model. So we are classifying, is this paper talking about a biodata resource or not? And this is a BERT model fine-tuned on a sequence classification task. And once, if a paper, passes this stage, then we're passing it through our second model, which is an NER model. It's also a BERT model fine-tuned on the NER task. And this is what extracts the actual names as well as acronyms of the databases. So for the paper classification task, our data set, we have a total of 1,500 papers. 400 of them have a positive label. 1,000 of them have a negative label. This is a fairly small data set because one of the challenges we have is for a lot of the things we're interested in, there are no openly available data sets, so we have to build them in-house. So in fact, this training data set has been curated by our collaborators at the Global Biodata Coalition. The best model, so yeah, that should say paper classification model, not NER. But our best model, we've experimented with multiple. It's a Roberta model. It's a Biomed Roberta model, and again, really impressive kind of metrics: 97 precision, 92 recall, and for a score of 94, F1 score of 94%. The way we're building this model here, we have access to the title and abstract of a paper. So we're concatenating this title and abstract, and then we're treating this as a sequence, a string sequence. And then similarly as before, we're passing this through the BERT tokenizer, and the fine-tuning part is this linear classification layer on top, where this is really just a sequence classification. So this layer classifies this string as either a 1 if it's about a biodata resource, or a 0 if it's not. For the NER task, we have our training data set is coming from 300 papers for covering a total of 1,500 biodata resource mentions. It's similarly curated by our partners. And here the metrics are a little bit on the lower side, and this is the best we were able to get. The best model, which was still a Biomed Roberta model, was obtained a precision of 71%, recall of 83%, and F1 score of 76%. Another thing, interesting thing we noticed is that, and this is why we kind of like always experiment with a number of different models, is that the best model on each task, on each of these downstream tasks, might be different for us. So previously it was a Cybert model, here's a Biomed Roberta model. So what we usually do is we just test all of them, and we've worked with them enough by now to know how to do that. And then we just take the best performing one. And similar as before, it's the same kind of architecture where we're concatenating the title and abstract, and then we're passing this through the model architecture, classifying each of the tokens as either being part of a resource or not. Here we have so right now we only have one tag for something being part of a resource, or like I guess one type of label. For something being a token being part of a resource or not. So basically we're putting together the full names and acronyms under one label. We've experimented with splitting them up, being able to predict, is this a full name or is this an acronym? And that showed a little bit of improvement in results, but it wasn't that substantial. The last thing I want to talk about is one of our projects to put together an open source data set of software mentions from the biomedical literature and making it available to the community through an open access license. So so far, and maybe you can see a team by now, a lot of the things we're interested in is surfacing these outputs from papers and making these links available. So in this case, we want to surface software from papers and make these links between papers and software available to people. And software is notoriously just not cited properly. I think around 31 to 40% mentions of software are formal citations, but that's also probably a bit inflated number, because a lot of the times people just don't properly mention the software they're using, which means it's hard to give proper credit for impactful software. So here we have an example where someone mentions in their paper that they used ImageJ to, draw an outline of whatever the region of interest was. So ImageJ is an imaging software. We would want to be able to say, okay, well, this paper used ImageJ, so we want to be able to surface this link. In the second example, we have an example of someone mentioning they utilized the Napari image viewer. So Napari is actually an imaging software that we're building in-house at CZI, and similarly, we want to be able to extract this link. So we also have a named entity recognition model here to extract these mentions of software, and I'm jumping a little bit here, but one particular challenge with software was that a software can really be mentioned in a paper under many different variations. So people have many different ways of saying the same thing, which is something we already knew, but we didn't expect with software to be so many variations. And there's also lots of variations coming from how the NER model is extracting these mentions. So what's in purple here are mentions extracted by the NER model. You have the obvious scikit-learn, but then you have the scikit-learn API, Python toolbox scikit-learn. You can have typos or differences, in how people refer to scikit-learn. And then you can have things like sklearn, which is just a different name for scikit-learn. So the challenge here is that if we want to look at the impact of software, we want to be able to count all of these as mentions of scikit-learn, because they should all be counted towards kind of looking at the impact of the scikit-learn software. So how we're doing that is we build the similarity matrix where really we're using three kind of like data sources, including what we call keyword synonym generation. So this is just as kind of saying, well, it's scikit-learn. If I see something like scikit-learn Python package, then that's just scikit-learn. Scikit-branch is a repository that's able to give us these mappings between things like scikit-learn and sklearn, knowing this is really the same software. And then we're also using string similarity algorithms. So we combine all of these things and we build this similarity matrix where really an entry in this matrix is a similarity score between, so A of I and J. This is a similarity score between string I and string J, where these are extracted by the NER model. So if the NER model has something like scikit-learn and scikit-learn Python package, then you'd expect these to be the confidence score to be 99% or something like that, because they're really the same thing. So we have the similarity matrix, and then what we do is we break it down into into connected components, and then we run the dbscan algorithm. So this is a hierarchical clustering algorithm. And we run dbscan on each of these connected components, and this is how we cluster these mentions of software under software entities. And it's interesting. So you can see an example here with SPSS, where you have things like SPSS statistics and also the full name statistical package for the social sciences. And actually, after we ran this clustering algorithm, we've discovered that SPSS had maybe more than 1,200 different software variation or string variations that were mapped under the SPSS software entity. So we ran all of these algorithms on 20 million papers, and we got a total of 67 million mentions of software. That this is before disambiguating them, and we made all of this data available. It's under a CC0 license. So I'm getting towards the end of my presentation. Just as a recap, the types of research outputs we've been mostly interested in so far are data sets, biodata resources, and software. And I want to end with some lessons learned. So we tend to deal with pretty niche tasks where we don't really have available data sets, which means a lot of the times we actually have to curate them from scratch. Luckily, we do have an in-house curation team, and we have the opportunity to partner with domain experts, and that they can help us with building these training data sets. When we're building the training data sets, entity definitions is one of the very first things that we do, and we learn they are very important because it defines what gets into the data set, but also it helps with evaluation. So we never deploy one of these models without doing some manual or qualitative evaluation. So then, which also involves our curation team. So if you have these entity definitions, you're able to say, well, this should or shouldn't be here. The scale we're working with, millions of scientific articles and the variations in natural language make it hard to use rule-based approaches on the things we're interested in. So this is where large language models really come to the rescue. We've also learned that just domain knowledge expertise is very valuable for us. So, people with biomedical knowledge or that have just deep knowledge in data set infrastructures and things like that. And last but not least, hopefully I was able to give you a few examples of using these large language models for good, where really what we're trying to do is using them to surface these impactful research outputs. And the end goal here is really to be able to find these impactful research outputs so we can keep funding them and give their creators proper credit. So thank you. These are our handles. If you're interested in checking us out, please do so. CZI Science, CZI Science Tech. The projects I've been chatting about today are part of our open science program. So feel free to check us out as well. And if anything, any of what I've talked about sparked any interest, we're always looking for collaborators and people to partner with. So if that's the case, feel free to shoot me an email, or you can find me on LinkedIn. I'd be happy to connect. And yeah, I think that's all I had.
Awesome. Thanks a lot. Anna Maria. Neeraj, I saw questions from you. Do you want to ask them directly?
Yeah, I was asking some of these questions very early in the talk, so I'll just summarize, maybe just ask everything as one question. I guess the first thing was for training your models, like are you using citations or other semantic information like author affiliations, author names, like, stuff like that, or journals in order to sort of enrich how you, or like set up how you train the models? And then the other question was how do you deal with diagrams and images inside papers? Those were the.
Yes, so it depends on the task. For things like extracting data sets and software, we don't really look at anything beyond the sentence. So what gets put into the training data set is really at the sentence level. So we're parsing all of these documents like scientific articles, and we're just getting to the paragraph level. So none of that information about authors or affiliations gets into the training data. And to your second point, we don't really include other types of like different modalities for data outside of text so far. So we don't really deal with, outside of maybe captions, we'd be able to, if there's a caption for a figure, we'd be able to mine that. But yeah, we haven't integrated those yet.
Harris, do you want to ask your question? I'm going through like from early to latest questions. Okay, yeah, go ahead.
Yeah, sure. Like a follow-up on that. So is it like the entire paper that you try to parse out the information, or just the abstract of the paper?
Yeah, it depends. Usually we try to do the full text, but it's complicated because there's only a small subset of the whole scientific, I guess, like ecosystem that is open access or we have access for. So we do have this in-house corpus at CZI that we have publishers agreement. So we have kind of access to this data. Even that is not complete. So usually what we do is we will use this corpus of full text we have in-house and run it on it, but we know it's not complete. And then there's these open access corpora, which are a little bit smaller. However, if you decide to run it on just abstracts, you usually get more data. So I would say it's a trade-off. Ideally, you want to go to the full text, but there's like some challenges with that. Yeah.
Thank you.
Shane, do you want to ask your question?
Oh, I saw that you were using semantic similarity to like match software names and stuff like that. So I'm sure you're doing that. But is there any demand for, I know I have this paper. Can I find similar papers?
Yeah, I mean, so we haven't worked on that specifically because I guess it didn't come up, but I can see how we can incorporate it, like, how it can be a subtask of other things we're interested in. So some things we're interested in, for instance, would be getting all of the papers related to a particular disease, or all of the papers related to a particular treatment, or things like that. So it could come up in that way. Yeah, we haven't kind of like built a model for that per se, but yeah, I can see it being integrated in other types of things we're interested in. Yeah.
Actually, maybe I missed that question about diagrams in the papers. Like, how do you guys deal with it?
Yeah, so far we don't really incorporate that type of data outside of just being able to look at the captions of the diagrams, maybe or parsing tables. So right now we're only focusing whatever is text, we'll be able to capture. So if it's a table with text inside of it, then yes, we'll be able to capture that. However, we're not incorporating any kind of computer vision or other types of things to parse imaging data or other type of data. Yeah.
I see a follow-up question from Shane. Are you guys translating non-English papers, or you're just focused on English
Yeah, that's an interesting question. I would say the corpora we use, it's mostly focused on English. Yeah, I don't think we have, and I think that's just by nature of how these biomedical repositories work. They don't think really include other types of languages to begin with. But that is a very interesting, yeah, point.
Yeah. Do you guys have like GitHub link, some like open source projects that our community can check out?
Yeah, I'd be happy to share those. Maybe, yeah, I can put them in the chat or on Slack after the meeting. Yeah, I'd be happy to share those.
Yeah, would be great.
Mhm.
Okay, I see a question from Eric. What are the key use cases for applying these LLMs?
Yeah, so sorry. I would say, let me go back. So the main thing we're interested in as a philanthropy, we want to recognize these outputs, impactful outputs like data, software, databases. So that's why we build these models to extract these outputs from scientific articles, because ultimately we're interested in looking at what are the software or data sets that have the biggest impact on science. And then we also want to be able to recognize these authors, people who are building or creating these outputs, and give them proper credit. So that's giving them proper credit. It's part of why we make all of this data openly available so they can use it and use it as part of accessing funding opportunities or for their own use cases. And I would say like the last but not least, we are serving researchers. So we are working closely with biomedical researchers, and it is part of our main, I guess, user population. So they're interested in surfacing these outputs. They just want to know what data set should I use, or what's the most impactful data in my data to use in my field or for my task. What is the most used software in my field? So questions like that. So I would say these are kind of like the three categories of things we focus on. One, it's internally for us as funders, we want to know what's impactful. Authors, we want to be able to give them this information so they can use them for themselves. And then we also want to serve researchers and make this data openly available to them. Yeah.
I see another question from Neeraj. Hold on. Do you think using semantic information like authors, affiliations, journals could make the models better?
I think it depends on the task. One thing we've learned or like we came across in the past is that so for the biomedical field or science in general, you tend to have these also co-citation clusters or just the same institutions kind of like citing each other, or the same labs citing each other. So in one sense, I think including that information might bias the models in some way, because you're including this other type of like potentially biased information where you might just end up with the most famous people or the most famous labs, things like that. So that would be my answer to that.
Mm-hmm. Eric, do you want to ask your follow-up question just to make sure that I understood it correctly?
Yeah, sure. I was just wondering, this sounds like almost like kind of like a Google search type of application, but I was wondering if you're for looking for articles that might be relevant to your research, but I was just wondering if you're planning to extend it to be more like ChatGPT, where you can just ask questions about the content and get answers.
Yeah, so it's something we're currently exploring. How can we use, something like ChatGPT, or I guess the types of things that people are using GPT for our own use cases. So we're currently looking into it. We haven't done anything in that realm yet, but yeah, I mean, when you're thinking about extracting information even from multiple papers at once, that's something that we'd be interested in and looking to explore. So, short answer is TBD. Yeah.
Okay, thank you.
Rahsan is asking, do you process documents per sentence or using a max length? What is the reasoning behind your choice?
So for training, we do have a max length for, I guess, let me go back. So let's say here where we're concatenating the title and abstract, this will have a max length. I think I have to go check, but maybe something like whatever, 512 or yeah, something like that. Whatever the maximum would be for a particular model. So it is possible some parts of the abstract are getting cut down. When we're running, so this is just for training data. When we're running them, so at prediction time, then we're not cutting everything. We're running on everything in the document. Yeah.
Mm-hmm. I'm wondering, like you mentioned that you had this initial training corpus. Did you guys label it internally, or you collaborated with some experts, domain experts, to build up that initial training data set?
Yeah, so what we usually do, we do have a curation team internally. In some cases, we'll try to get the data if there's some things available. And because I mentioned we have these kind of like very niche cases, we almost always will have to curate it. So usually the way it works, we look around, we see what's available. If someone else has done something similar, we try to start from there so we don't start from scratch. But we use our curation team to kind of refine the labels or, filter the information in some way. So we're able to apply it to our own task. Our curation team has around 10 people, so it's fairly reasonable, but still it can't scale that much. So we prefer to start from something, not from scratch. In this case, so for this task, our curator, this was actually a trained data set, a data set put together by our external collaborators. So you can see the size is pretty small, right? 300 papers. It's kind of small for a data set to use for our tasks. So that's this is where we started from scratch. That's one of the downsides of starting from scratch is you're going to end up with less data. Yeah.
Yeah. Do you guys have other questions?
Can I just ask? So, I think you made a very good point that like if you use things like affiliations and so on, it could actually introduce bias into the data set. But oftentimes that kind of information makes it into the text of the paper itself, right? Like in the form of acknowledgements and stuff like that. How do you prevent like that kind of leakage of bias into the data?
That's a great question. Right now, we don't really do anything, but that's a great point. It's something that we'll have to, we actually haven't considered that necessarily, that it might leak in that way, but yeah, that's a great point. Yeah.
Eric is asking, do you have an idea of the cost of preparing these models? Do you mean like these models, Eric?
Oh, just how much for the compute time, like how much computing resources it required and what that just like an order of magnitude or.
Yeah.
Yeah, so we do have access to GPUs. I know if people maybe work with or are familiar with Databricks, we do use Databricks to train these models. This is just cloud computing. I will say that, again, I think it depends for this one in particular, where it's a fairly, we have fairly small data. It will probably finish, if I remember correctly, in like 1 hour. So that's okay. Some of our more bigger models. So if we go back to this one where we have 120,000 sentences that have to go through the model to learn from, I think it's actually took us a solid few hours, if not a day, to train. I don't know the exact cost of it, what's involved, GPU per hour, things like that. But I would say, yeah, it depends. A couple of hours to maybe like 1 day-ish.
Okay. Yeah, very good. Thank you.